All right, as we were just talking about, a eukaryotic cell is a little bit more complicated. As you can see here is a cross-section of eukaryotic cell, and well, one thing is for sure, we've got a ton more stuff that's presented. All of these different membrane-bound organelles. We've got, I'm going to highlight this in the middle, we've got our, we've got our nucleus, this whole thing right, this sphere in the center. In the center of the center is the nucleolus, and within there is our chromosomes, our chromatin, our proteins that are our genetic material that is kind of wrapped up in our wrapped up in a bunch of proteins. We've got this nuclear envelope, which is kind of the sac that the nucleus is found within. And as you look right there, there's little holes, there's little pores within that nuclear envelope. So if you go out from the nucleus, one of the first things that you should see is this guy right here and this guy right here. Well, what these are the rough endoplasmic reticulum. And it's a rough endoplasmic reticulum because of the fact that we've got these little blobs, these little dots on the surface of that rough endoplasmic reticulum, and those little dots are ribosomes. So ribosomes are on the surface of the rough endoplasmic reticulum. Okay, now if you go out from the rough endoplasmic reticulum, you have something that's similarly in this kind of weird, like, I don't know, it almost looks like a bunch of pancakes. We've got the smooth endoplasmic reticulum. Now, the difference between the rough and the smooth endoplasmic reticulum from a visual standpoint is the smooth ER, which it is also known as, does not have any ribosomes on it. Now, outside of those two regions, we've got a couple of other players. We've got the lysosome, these little blobs right here. We've got the mitochondria, this guy right here and right here. And then we've got centrioles, which are involved in cellular division and replication. And then outside of that, we've got this other kind of pancake type structure known as the Golgi body, also known as the Golgi apparatus. We're going to go into most of these different components all together in just a little while, but the key is we've got a bunch of membranes within our cell, within the cytoplasm. The outermost layer of our eukaryotic cell is our plasma membrane, and the surface of that is our cellular coat. Okay, so eukaryotes, we've got a plasma membrane. It separates the cell from our external environment. There is no cell wall in a eukaryotic cell. Now, if you talk about a plant, well, plants do have cell walls, but they're kind of the dividing factors that keep. Seems kind of weird, but they don't go with the plasma membrane in some respects. So the plasma membrane, what is it made up of? It's made up of a bunch of lipids and proteins. It's going to control transport, it's going to provide shape and structure, and it's going to play a role in intercellular communication. It's going to allow cells to respond to external stimuli, as well as provide a means for one cell to tell another cell something else. For instance, if there's like a process of autophagy taking place or a process of senescence taking place, well, it's that the plasma membrane is going to mediate those communications between cells. Now, the nucleus is going to contain the cell's genetic information, your DNA. It's going to play a role in metabolism by influencing the rate of protein synthesis. It's going to be surrounded by the nuclear envelope, and that nuclear envelope is going to kind of serve analogously to the plasma membrane in the sense that it's going to separate processes in the nucleus from the cytoplasm. This is going to allow for more sophisticated regulation of gene expression. So this is going to kind of tightly control which genes are turned on and which genes are turned off, and when they need to be turned on and off. The endoplasmic reticulum, we had two examples of these, the rough ER and the smooth ER. The rough ER, as I said previously, has ribosomes on the surface. It looks rough. That name is dated back to its appearance, or dated back to any sort of microscopic investigations of the rough ER. This is going to process membrane proteins and water-soluble proteins destined for the endoplasmic reticulum, other organelles, or outside of the cell. Okay, so key there is the proteins that are synthesized at the rough ER, where do they go? They go to the membrane, they go to the smooth ER, they go to other organelles, and they go outside of the cell. Now the smooth ER, there are no ribosomes on it. The smooth ER is responsible for lipid synthesis, biotransformation, so water-insoluble organic molecules are prepared for excretion, and calcium storage. Now, the lumen is the space within the endoplasmic reticulum. This has a list of all of the different diseases or disease states that are related to endoplasmic reticulum stress. Alzheimer's, Parkinson's, type 2 diabetes, atherosclerosis, sorry about that, non-alcoholic fatty liver disease, as well as alcoholic liver disease. So disruptions and stresses on the endoplasmic reticulum can lead to a very wide range of disease states. Now, as it comes with eukaryotes, we've got our ribosome, which we mentioned a little while ago. The ribosome is this was our self aggregation, where we have RNA and proteins coming together. And this is the site of protein synthesis. This is not an organelle. It's going to be found on the rough ER, free within the cell, or in the mitochondria. And this is something you might think, well, that seems a little bit odd, but well, that's because the evolutionary history of the mitochondria. The evolutionary history of the mitochondria is that it was its own standalone organism, or it's theorized that it was its own standalone organism at one point in the past. So it has what that means is it has its own genetic material, it has its own means of basically surviving. So it's a symbiotic relationship between a eukaryotic cell and a mitochondria. So our ribosome is found on our rough ER, free within the cell, or in the mitochondria. Is it found in the lysosome? Is it found in the smooth ER? Nope and nope. Now, here's a nice little depiction of what that looks like. We've got these little spheres, which are our ribosomes. They're floating around, or they are on the rough endoplasmic reticulum. Now, a visual of what this looks like, well, it's depicted right here. Here we have our 5' messenger RNA termini, and it goes to basically the center point of the large and the small ribosomal subunit, and it's there that a polypeptide is synthesized and is produced. Now, ribosomal disorders, also known as ribosomopathies, probably pronounced that, well, I pronounced it as well as I could. They do exist. I would speculate that if there are gross disorders, well then that it's going to be something that prevents an organism from really developing at a very, very early stage, because proper functioning of a ribosome is an essential part of survival. But these two disorders that we're going to mention are Shwachman-Diamond syndrome and Diamond-Blackfan anemia. So, as you can see, approximately 1 child in every 100,000 is born with Shwachman-Diamond syndrome, and 5 out of 1 million have Diamond-Blackfan anemia. They have some pretty significant differences in their outcomes in terms of what happens. But what Shwachman-Diamond syndrome leads to is a child's pancreas does not release enough enzymes in order to digest fats, resulting in fatty bowel movement. Now, Diamond-Blackfan anemia, they have this because the bone marrow is not making enough red blood cells. This disease is due to mutations within a total of 7 genes. Some of these genes give instructions to the cell on how to actually make ribosomal proteins, while others help the ribosomes make proteins altogether. Now, our next organelle within the eukaryote is the Golgi apparatus. This is a flattened sac-like vesicle. Slide here says it looks like plates. I was going with pancakes. But here is our Golgi apparatus right here. And what you can kind of see here is what happens to a protein that is synthesized at the rough ER, or sorry, the smooth ER, which that can be transported within a vesicle to the cis side, the same side as the nucleus of the Golgi apparatus, at which point this vesicle will I guess it will join up with or combine with the cis side of the Golgi apparatus. These proteins that are packaged within this vesicle will be processed and ultimately packaged into secretory vesicles, and those proteins will be transported out of the cell altogether. So the Golgi apparatus is a means of exocytosis, or it's a means of it's a means of transporting a protein outside of a cell. Now, exocytosis, exo meaning out, and cyto meaning cell, exocytosis is the secretory process that involves fusion of membrane-bound granules with the plasma membrane. So basically we're packaging up a protein in the smooth ER, transporting it to the cis side of the Golgi. It's being processed through the Golgi, at which point it's blebbing off and forming this secretory vesicle that goes on to merge with the membrane. So these proteins, one of the important things about them is proteins that are exocytosed, or molecules that are exocytosed, they don't ever really see the interior of, or they don't see the cytosol, because of the fact that they are constantly kept within a secretory vesicle or a transport vesicle. So there you go. Now, there are a number of different neurodegenerative diseases that are linked to malfunctions of the Golgi apparatus. So misfolding of proteins, you can also see disruption of calcium homeostasis, because if you remember, one of the things that the Golgi does is it is involved in calcium storage. Neuroprotection of hormones. It can also be an insult from a Golgi apparatus malfunction. Lysosomes, one of the other organelles. So we've talked about the nucleus, we've talked about the smooth and rough ER, we've talked about the Golgi apparatus. Now we're going to go on to the lysosome, which is a single membrane sac-like organelle that contains aggregates of digestive enzymes. Now this is at a low pH. This is approximately 5.5. If you compare that to the cytosolic pH, which is about 7.4, well, those are pretty significantly different environments, especially when you consider the fact that our pH scale is a log scale. So it's a pretty big jump from a pH of 5.5 to a pH of 7.4. Now, this exemplifies the importance of compartmentalization because of the fact that we can have this entity within a cell with a really low concentration by contrast to the environment around it. The lysosome degrades debris within the cell through a process known as autophagy. It's going to play a role in receptor-mediated endocytosis, so bringing something from outside of the cell in, and then it's involved in a number of different secretory processes. So these digestive enzymes in the lysosome, they're only going to be functional at a pH of 5.5. Because if you move them out of there, well, their activity is going to just plummet, because that's a really distinctly different environment. From that pH of 5.5, it's a pH of 7.4. Now, the lysosome can malfunction. In some people, there are genetic mutations that cause deficiencies or malfunctions in one of these lysosomal enzymes. Molecules that would normally get broken down instead accumulate. So if you have lysosomal, so our normal function is this complex substrate goes into the lysosome and gets broken down. So we've got these different shaped objects, this red, orange, blue. Well, in enzyme A, it's going to take off the blue components, break that down. Okay, so now we're left with the orange and the red components. They're stuck together, and our next enzyme is going to pick off all the orange ones, and then our next enzyme is going to pick off all the red ones. And so we've taken this complex mixture and we've broken it down. And those end products can basically diffuse out of the lysosome and go out into the cytosol and basically do nothing significant. Now, in the event that you have, or there are some instances where lysosomal enzymes are malfunctioning or not properly functioning, in which case you see step 1 do its job, but then step 2 doesn't do its job. And so you've taken this complex structure or this complex substrate and you make it a little bit more simple, but you haven't broken it down enough that it can diffuse out and be processed down farther. And so then you just have these things that aggregate and accumulate, and ultimately that's going to be potentially catastrophic to the cell. So an example of this is Tay-Sachs disease. This is a lysosomal disorder. And this is responsible for ganglioside breakdown. Right here is a ganglioside. So enzyme responsible for ganglioside breakdown misfolds, so it's not in its proper 3D shape, and it's destroyed. Usually affected infants seem normal for the first few months. However, when this begins to be a problem, thinking about it in terms of an accumulation, these gangliosides accumulate at higher and higher levels inside their nerve cells. Within 3 to 6 months, the child becomes irritable, listless, and may actually develop seizures. Blindness, deafness, and paralysis ultimately follow, and usually this is something that is it's generally fatal by the age of 5. And so if you think about when something accumulates, it's not going to be fully processed, and so it's just going to, like, you know, if you think about any sort of stoppage and accumulation of something, it's ultimately going to reach a breaking point unless there's some relief that's provided. And unfortunately, Tay-Sachs disease is one where you can't really provide that relief. Here's an example of what a ganglioside looks like. We'll talk about these a little bit when we talk about lipids, but a ganglioside consists of 3 major components. We've got a sugar component, a fatty acid component, and a sphingosine component. So fatty acid, sphingosine, and sugar. And that's ultimately what a ganglioside looks like. We'll talk about these later when we talk about lipids, though. So these are found in the exterior of the cell, and they are commonly used in communication. So if these are on the cell surface, they provide a means to communicate with an external part of the cell, or an adjacent cell, I'm sorry. Next up is our peroxisomes. These are small spherical membranous organelles that contain oxidative enzymes. These enzymes are involved in catabolic and anabolic processes, that produce peroxides. This exemplifies the importance of compartmentalization, just like the lysosomes does. So when you think about compartmentalization, keeping these unique environments separate from one another, your hallmarks, I think, are it's almost like everything else does good work, but the lysosome and the peroxisome prevent bad things from being found within the cell. So this separates our toxic environment from the rest of the cell. So peroxisomes and lysosomes they just do some fantastic heavy lifting to keep the bad stuff from going everywhere. Now, there are a number of different peroxisomal disorders. Zellweger syndrome is an example of an absence of function of the peroxisome, leads to the accumulation of long-chain fatty acids within the brain. So think about that as one of the things that peroxisomes do. They break down long-chain fatty acids. And so what this also leads to is the decreased formation of plasmalogens and defects of bile acid formation. So those are 2 parts of ultimately fatty acid breakdown and proper processing of fats. So that's one of the implications of peroxisomal disorders. Next up are mitochondria. which if you've ever taken a high school biology class, you probably all got to say, what's the mitochondria? It's the powerhouse of the cell. It's also known as the power plant of the cell. I'm accustomed to powerhouse, but power plant works just as well. This is responsible for aerobic respiration and ATP synthesis. This is a complex organelle containing a pair of membranes, an outer porous membrane and an inner impermeable membrane. So what the difference between these two things is that porous membrane, there are some things that are smaller than or equal to 10 kilodaltons. So this would be an amino acid, or sorry, this would be a protein of, say, 100 amino acids that can migrate in and out of that porous membrane. This inner impermeable membrane is going to prevent small ions from transporting across that membrane. So the outer membrane, fairly large things can jump in and out of it. The inner membrane is very, very small, and it's going to very tightly control what migrates in and out of it. Now, the space between the inner and outer walls, or inner and outer membranes, is known as the intermembrane space. Now, this is a very key part of where oxidative phosphorylation takes place. We'll talk about oxidative phosphorylation later on when we're talking about, well, ATP synthesis and well, our last bit of central metabolism. Now, the space within the inner membrane is called the matrix. So this is going to contain several circular DNA molecules and components for protein synthesis. So within the inner membrane is where DNA is going to be stored. And that DNA is going to be key for synthesis of proteins that are involved in mitochondrial function. Because if you remember, I mentioned that the mitochondria evolutionarily was its own organism. Now here's a visual of what we're talking about. So we've got the mitochondrial matrix right here. We've got this inner membrane, which is indicated by this, I don't know, kind of khaki, light brown. And then the outer mitochondrial membrane is this outer, or this darker membrane there. And so there's a nice picture there, but an electron microscope image is displayed here to the right. So these depictions, you know, it's definitely a simplistic view of it. But we'll talk more about the mitochondria whenever we're talking about oxidative phosphorylation specifically. A number of different mitochondrial disorders are present, which, as you can imagine, if it's the powerhouse of the cell, well, if something goes wrong, it's going to be potentially catastrophic. So mitochondrial disorders in different tissues have different results. So optic neuropathy, for instance, is a disorder that has to do with mitochondria within eye cells. And so you have all these disorders that are likely the result of ATP synthesis, in which case if the mitochondria is not functioning, or sorry, one of the ways in which the mitochondria is not going to function properly is it's not going to be able to synthesize ATP sufficiently, and that's going to lead to a breakdown of tons and tons of muscular systems. So as you can see, mitochondrial disorders are linked to a number of different diseases in different tissues. So the cytoskeleton, originally thought as a structureless solution, it was just, oh, well, you know, your plasma membrane was basically like a Ziploc bag, and the cytoskeleton, or the cytosol, was just the solution within. Well, actually, not quite. The cytoskeleton is a network of proteinaceous fibers and filaments. So you have these networks of proteins that it's almost like if you drove through like not necessarily an older city, but if you drove through a city where there's tons of power lines up, that's almost what it would look like. If you look over in the lower right-hand corner of this, this slide, this shows filament proteins that have been tagged with the green fluorescent protein, and so you have this very complex network of proteins that are going all over the place in the cell. And what they're going to do is, one, they're going to provide shape for the cell. So this network has basically two ends on, there's one end anchored over here, another end anchored over here, and you have all of these different filaments. But it's also going to allow cellular movement. It's going to allow the cell to move, but it's also going to, the cell to move, because these threads, these strings can contract and expand, expand. But this is also going to provide supporting structure for organelles. So if you look in the lower left-hand corner, what we've got is we've got these microtubules and we've got these microfilaments of proteins, and those are basically serving as support for our mitochondria and our endoplasmic reticulum, and outside of that is our cellular membrane. So many diseases now have been associated with abnormalities in cytoskeleton and nucleoskeletal proteins, including a number of cardiovascular diseases, cancers, liver cirrhosis, pulmonary fibrosis. Basically think about it as, if it's not the right shape, it's going to run into some problems. And so the cytoskeleton is going to dictate the shape of that cell, and if the cytoskeleton isn't, properly functioning, well, then it's not going to be the right shape, so it's not going to necessarily carry out the right function. How do cells communicate with one another? Whether or not they are unicellular organism or found as a part of a multicellular organism, cells are going to adjust within their environment and communicate with one another. So in some cases, if you have in the upper left-hand corner here, we've got contact dependence. So a signaling cell has a membrane-bound signal molecule that comes in contact with a target cell that has a membrane-bound receptor protein. So you have that direct contact between the signal and the receptor. We also have a paracrine system, in which case a signaling cell is going to release something that is perceived by target cells and their receptor proteins. In synapses, you have a neuronal cell that has a cell body, an axon, and then a neurotransmitter region, which is going to release those signaling molecules that come in direct contact with those target cells. And then in endocrine cells, you have a hormone that's released from the endocrine system. An endocrine cell finds its way into the bloodstream, moves throughout the bloodstream to then hit the target on that target cell. So we have direct contacts, proximal contacts that are with mobile signals, and then distant contacts with mobile signals. And then the synaptic communication is, well, you have a cell that's a unique shape that has a region that is where the communication takes place. So to illustrate how a cell responds to a stimulation, we'll use insulin as our model. Now, anytime that you look at any sort of illustration like what we see here, it's important to remember a couple of things. Think back to earlier on, I said cells are really complex, and we had this very narrow view of, okay, well, this molecule does this, that allows this into the cell, and then that's the response. Well, we've got that same sort of thing here, but even this, it's like, I've been narrowed down, but this is complicated. And if you're looking at this and thinking, oh, this is kind of complicated, where do I start? Well, what I would advise is, I guess there's part of me that's like, well, you got to start somewhere. But the way that I always like to think about insulin and diabetes is I think about sugar and I think of insulin. Those two things. I want to bridge the gap between those two things. So what we've got here is a cell that has a GLUT2 transporter protein. Now what that protein does is it opens up and allows sugar to come into the cell. Okay, so let's look at this. So we've got, in this corner, we've got high glucose concentration. Now, GLUT2 is going to open up and it's going to bring individual glucose molecules in. Those glucose molecules will be processed and they will make a molecule known as glucose 6-phosphate. that glucose 6-phosphate is going to go to the, well, spell it with me, mitochondria. Now, what that is going to do is ultimately lead to a production of ATP. And if you're looking at this and you're thinking, where did this all come from? This is not something I expect you to know this information on this exam, but this is stuff that we will ultimately get to over the course of the semester. Glucose 6-phosphate is going to be metabolized, and it's going to cause an increase in ATP concentration. What that ATP concentration is going to do is it's going to block potassium ATP dependent channels. So it's going to block this protein. Whenever that protein is blocked, that's going to basically lead to depolarization. So what that means is it's going to let ions from outside of the cell, where there's a higher concentration, flow into the cell. Now all of a sudden we have these calcium ions that flow into the cell. And when these calcium ions flow into the cell, they're going to activate different things. One thing that they're going to activate is an enzyme known as ERK1/2. What that ERK1/2 is going to do is it's going to activate a transcription factor that's going to it's going to initiate the translation of insulin. Now, insulin is going to be packaged up into a transport vesicle. It's going to go on to the membrane. It's going to open up, and now all of a sudden, insulin has been released. Okay, so that's our very simplistic flowchart of what happens. Now, is that the whole story? No, it's not. We kind of took a very quick roll through this, and we're not, at this point in time, I don't want to bombard you with everything else that's at play here, but as you can see, the presence of calcium right here leads to other things. There are other components that lead to ultimately, it's almost like this is a maze, but there's 7 ways to get to the destination, or to get out of the maze or into the maze. I can't remember how to do a maze. Through the maze, there we go, that's the word. So this is what insulin gene transcription in beta cells looks like. It's very complicated, but that's essentially a functioning, or a proper functioning of things. And as you can see, we've got mitochondrial influence, we've got transport and exocytosis of proteins, or exocytosis of insulin. So we have all of these different components at play that, well, as you can imagine, are going to influence a cell's or a person's response to high sugar concentrations. So we just looked at how insulin is produced through sugar coming into a cell. Now we're going to look at, well, once that insulin is produced and released, what is the fate of that, and what does it do? Well, insulin would be an example of one of those mobile signals that is bound ultimately to a receptor protein. That receptor protein has a region that spans the plasma membrane, so this is outside of the cell, as is indicated right there. And insulin is going to cause an effect on the interior side of the cell. And so when it's received, then the cell undergoes, proteins within the membrane, and then ultimately within the cell undergo a number of different conformational changes, and basically a response is stimulated all across or within the cell. So this insulin does not have to make its way in, and all of this is mediated through the presence of the plasma membrane. So essentially, there are some molecules that can go into that plasma membrane, as was indicated by our glucose, because there was a protein that opened up, glucose flowed through, and then you saw this cascade of events that took place. Then there are other molecules that do not need to actually enter the cell in order to result in their change, or produce any sort of change that they're going for. All right, well, this is going to wrap up chapter number 2. This is a lot of information. What I want you to take away from this chapter is, what are those major organelles, and what do those organelles do? Then I want you to think about, well, if something is wrong with one of those organelles, what is possibly the outcome of that? And so I hope this is helpful, and I'll see you in chapter 3.